Excited to have, oops, sorry, to have this opportunity to talk about traits and some of the research in my lab. And I'm excited to be presenting it to GLION and students in particular, because I think GLION is a really a wonderful organization because it's grassroots. It includes scientists from all over the world. So I'm really excited about this. So I'm going to talk about trait-based aquatic ecology, and I will focus on phytoplankton because that's what I mostly study. But while I'm giving my talk, I think you can think about your favorite organisms or favorite processes and kind of think how these ideas could be applied to your study system or to your favorite organism. So, as I mentioned, we study a lot of different things, but I think many aquatic ecologists are answering very similar questions. So one of the questions that interests me, and I'm sure many of you are pondering, is how do we explain ecological community structure and dynamics? For example, can we identify the mechanisms that structure aquatic communities? And then, of course, an important question is how do we predict what communities would look like in the future, especially with rapid environmental change? And there are many questions within this big question. For example, can we predict what species will dominate and under what conditions, or what species will decline? Maybe some of them will go extinct altogether. Then another interesting research area is how will communities and ecosystems and individual species respond to extreme events, either in temperature, precipitation, and other environmental factors. And the question that interests me and probably interests many of you in the audience are whether harmful algal blooms would increase in the future. And of course, when we think about the functioning of aquatic ecosystems, we need to know if major functions such as photosynthesis, respiration, biogeochemical cycling will change in the future. So all these questions are difficult, and I suggest that a trait-based framework could be such a mechanistic tool to address successfully many of these questions. And so I will present some of the examples from our lab, and I will talk about future challenges in my talk. So just to illustrate how traits can help us think mechanistically, we could go to the terrestrial realm and think about the primary succession in plants. So we all know from our high school or early university studies that the primary succession starts on bare ground, and first colonizers are mosses, lichens, and other similar organisms. Later on, the succession leads to the dominance of different species. And finally, we have shrubs and trees. And so there is this nice sequence of dominance of species. And so when we think about this sequence, it's hard to understand what mechanisms drive this succession. So I suggest that when we look at traits, we get a much better understanding of the mechanisms that are responsible for this process. For example, the early colonizers are known to be to have high light requirements, so they're poor light competitors, but they're good nutrient competitors. On the other hand, late successional species are good light competitors and poor nutrient competitors. So immediately from looking at traits, we can understand that nutrient and light competition may be driving the succession, with nutrient competition being maybe more important in early on, and light competition being more important later in the succession. So of course, traits can inform us about potential mechanisms. So I'm talking about plants, and it's not a coincidence, because trait-based ecology really became the mainstay in terrestrial plant ecology. And I wanted to show you how citations increased with time, citations that focus on traits and plants. So I started doing this search in ISI Web of Science, and it gave me this unsatisfying answer, so that my search found too many results. When I searched for traits and plants, okay, I decided to go narrower. So I entered traits in trees, still no luck, too many publications that deal with it. So then I decided, okay, I'll go even narrower. I look for traits and tropical trees. Finally, the ISI gave me the results. And you can see how the focus on traits really increased exponentially. So, for example, in 2008, there were 4,000 citations dealing with traits and only 75 papers, at least what ISI found. And then last year, it was many, many more citations and publications. So to return back to the aquatic realm, I decided to see how phytoplankton are doing in terms of the focus on traits. And I did it because trait-based approaches have been applied to phytoplankton for a while now. And that's what my primary interest is. So when I searched for traits and phytoplankton, I found that, well, yes, the mentions of traits and phytoplankton are increasing in the literature, but of course, we're nowhere near the literature, the amount of literature that deals with traits and tropical trees, for example. So why is that? Does that mean that traits or trait-based approaches aren't appropriate for phytoplankton? Of course not. It means that we still haven't really utilized all the potential that that we have in using trait-based approaches. And so the situation is changing. As you can see, the citations are increasing. And so I think trait-based framework is a nice mechanistic approach that would allow us to tackle all those questions that I formulated in the beginning and many others. So when we think about traits, we can go back and like how basically and look at early models of plankton, and both in marine and freshwater systems, plankton communities were modeled in a very simple way. So basically 20, 30 years ago, it was one box or one equation for phytoplankton, then for irradiance, light, and nutrients, and then zooplankton. So the system was easy, this model system was easy to investigate, but of course it was highly unsatisfying in terms of reflecting the reality of all the diversity. So later on, the models started adding diversity by including different functional groups. So I'm focusing on phytoplankton here. And of course, this is still not satisfying because there are many more species and functional groups. So how else can we do it? Well, of course, sort of the brute force approach would be to describe all the species of interest with their individual equations. But of course, this would get out of hand very fast, and parameterizing these models would be a nightmare. So trait-based approach takes a different sort of view of all the diversity. It looks at the continuum of strategies. So when you think about phytoplankton, you can think about different strategies in terms of light competitive abilities or nutrient competitive abilities, or along another dimension, you can think about grazer resistance or being a good competitor as another sort of axis for ecological strategies. So when we think about the continuum of strategies, it becomes easier. We don't need as many equations as we would if we were to model individual species explicitly. So for example, we also can think what conditions would lead to the selection or the highest fitness, and what strategies would have the highest fitness. For example, under low light, we may have a strong selection for good light competitors. And so we could then identify species that fall into this part of trait space. And then we could identify or predict what species would potentially be dominating under low light conditions. And similarly, under low nutrient conditions, we may identify species with high nutrient competitive abilities and then predict that they would be selected for. So this makes our predictions easier because we can sort of focus on traits instead of species and identify then traits that, species that have those traits. So as I mentioned, the advantages of trait-based framework is that it focuses on relevant traits, not species. So there is some level of abstraction there, but you can go back from traits to species if you wanted. Then trait-based approaches can help identify the mechanisms that structure communities. And I gave an example from the primary succession, and there are many more. So as I just showed, trait-based framework can reduce complexity while still retaining this diversity. And you can think about this continuum of ecological strategies. So this framework also helps link ecological strategies, functional diversity, and community assembly. So basically, I'm a strong proponent of looking at traits and trade-offs. And so we can define what a trait is, and there are many definitions, but here is one. So trait could be defined as any quantifiable characteristic of a phenotype or an organism, which is presumably governed by a specific gene or genes. And so there is another term that is frequently used, and it's a functional trait. So in other words, it's a trait that affects fitness. And so traits could be values or traits could be functions. Here is an example of a function valued trait, which is a thermal response curve. And so you can describe it as a function, and that could be viewed as a trait. Later on, I will show that there are other potential traits that could characterize this temperature response. So what are the components of trait-based framework? Of course, we need to know traits. And we also need to know trade-offs between traits. And as I showed earlier, those trade-offs can help us characterize different ecological strategies. And I will also give examples later. We also need to think about relevant environmental gradients. And for many species, that would be temperature, nutrients, predation pressure. And so we shouldn't forget about species interactions. And this is like basically the most challenging now. The most challenging aspect of trait-based ecology is to understand what traits determine these interactions. And we need to agree on how we can measure fitness. And for many organisms, growth rate could be a nice proxy of fitness. So as I said, I will focus on phytoplankton. And so what are the important traits in phytoplankton? So of course, it depends on the question that you're interested in, but often the important traits are cell size, and it is often called master trait. And so then the maximum growth rates, nutrient and light utilization traits, or competitive abilities for nutrients and light. Then temperature traits, grazer resistance, buoyancy, resting stages and dormancy, nitrogen fixation. And of course, this list is not complete. So you can also try and classify traits according to their ecological function and the trait type. So for functions for many organisms, not only for phytoplankton, we have reproduction, resource acquisition, and predator avoidance. And for trait types, we can distinguish morphological traits, physiological traits, behavioral traits, life history traits, and for a lot of microbes, it would be genomic traits and some other traits, metabolic traits. And so you can also, so that table was for phytoplankton. Of course, it was not filled completely, but here is our attempt to do a similar classification of traits for zooplankton. And again, body size transcends different ecological functions, as we all know. So next step in trait-based framework is to actually collect the information on traits, trait values, and to look at the extent of trait variation. And here is an example of the analysis of the compilation that our former postdoc Kyle Edwards did. And he looked at many different traits of phytoplankton. And here is a graph representing the distributions of maximum growth rates in different groups of phytoplankton, both for freshwater and marine environments. And as you can see, there is a lot of variation. So basically different species can have different trait values even within individual groups, such as cryptophytes, diatoms, cyanobacteria, chlorophytes, and so forth. But you can see that there are still differences in the means of traits for different groups. And so that means that different conditions may select for different groups of phytoplankton. And so, as I said, it's important to look at the correlations and trade-offs between different traits. And so here's an example of how the optimum irradiance for growth is correlated with alpha, the initial slope of the growth irradiance curve. And so basically high alpha means good light competitive abilities. So as you can see, there is a relationship between these two different physiological traits. And it seems that diatoms and dinoflagellates may have some differences as well. So when we think about ecological strategies aligned along this trade-off, so we can distinguish these two extremes, high light adapted ecological strategies and low light adapted ecological strategies. And of course, there are a lot of species in between. But you can see that there are differences in these strategies due to these correlations and trade-offs. So an interesting finding that we had several years ago, well, a while ago, is that traits are correlated not necessarily in a pairwise fashion, but also there are multi-dimensional trait correlations. And so here's an example of this three-way trade-off where we looked at the nitrogen competitive ability, phosphorus competitive ability, and cell volume, which is a proxy for grazer resistance. And so we can see that different species represented by these dots align themselves on this surface. Basically, it's a plane. And what was also interesting is that marine and freshwater groups occupied different parts of this plane, basically showing that there are different trade-offs important in two different environments, in marine versus freshwater. So for freshwater environments, we saw a negative correlation between nitrogen and phosphorus competitive ability, while in marine environments, we saw a positive correlation between nitrogen and phosphorus competitive ability. And there was a larger variation in grazer resistance in the freshwater realm, so which was also interesting. So we can look beyond the pairwise correlations. And of course, if you can think in higher dimensions, you can think about 4-dimensional trade-offs and so forth. that is an interesting exercise probably. So another question is, can we use traits to predict community composition and dynamics under global change? And the answer is, of course, we can. And so I will give you an example where we looked at temperature as a stressor. And we all know it's a global stressor for both freshwater and marine environments. But this first example is for marine environments. And so the ocean is predicted to warm. And so we wanted to know how this warming would affect different groups of phytoplankton and the overall biodiversity of phytoplankton. So what we did, we looked at temperature-related traits. And so, as I said, there are several traits associated with temperature responses. And so one important trait is the optimum temperature for growth. So, and the minimum critical temperature and the maximum critical temperature, and also the niche width. And so we collected these traits for a lot of different species isolated from different parts of the ocean, from the poles to the tropics. And then we use that trait information to predict the individual distributions of species in the ocean at present and in the future. And so we were able to see how thermal niches would change with warming. And so here are the examples for two species. So one species is a temperate species, a coccolithophorid, and another species is a tropical species, a nitrogen fixer, Trichodesmium erythraeum. And so in gray are the present day niches. And so in red, we show how niches would shrink in the future. And in blue, we show how niches would expand. And so for this temperate species, warming would lead to the shrinkage of its niche and closer to the tropics, because it would be too hot for the species to survive. And it would be moving polewards towards more growth conducive temperatures. So, and then for the tropical species, we predicted that its niche would expand because there would be more suitable conditions in the future for the species. So then we put all this together, the predictions for more than 150 species, and we assessed how diversity would change. And we found that the diversity is predicted to decline in the tropics. Mostly because a lot of species would be too hot. It would be too hot for a lot of species to survive. Sorry. And so in contrast, in temperate regions and closer to the poles, the diversity may increase because more species would be able to grow in those high latitudes. So these predictions were pretty stunning, but since then, there were studies that are supporting these predictions. And so it is a little bit counterintuitive to think that in the tropics, the diversity would decline. And even the sometimes it will be too hot for the tropical species to survive. So temperature traits are really informative. And we also looked at temperature traits in other systems. And here is an example when we looked at temperature responses of different phytoplankton species in Lake Baikal. So Lake Baikal is undergoing rapid warming. And at the same time, it's a home to a lot of endemic species, including endemic phytoplankton. So we isolated a lot of different species, including the endemic diatoms, and we measured their temperature traits. And we found that there are a lot of cosmopolitan species that occur in different parts of the globe. And so for the cosmopolitan algae, we saw that they can survive at really high temperatures that are never experienced in Lake Baikal. So for example, this species of green alga can grow well up to 25 degrees Celsius. And Lake Baikal can warm only up to the central Lake Baikal, the surface waters can warm only up to 15 or 16 degrees on really warm, during really warm periods. So basically, this cosmopolitan phytoplankton has the capacity to grow fast even in the warming climate. In contrast, we also looked at endemic diatoms that bloom under ice. And so here are two members of our Lake Baikal team, Kirill Shapov and Ted Oziersky, and they're using this like sort of low-tech way of getting samples on the lake in winter to get to those endemic diatoms. And so Kirill Shapov measured growth curves and temperature traits for this endemic species of this diatom, Olacosera baikalensis. And we found that basically this species is adapted to really low temperature regimes. So it cannot grow above 10 or 12 degrees Celsius, and its optimum temperature was around 4 degrees Celsius. So with warming, the potential for the species to survive is declining. And we had a model, mathematical model, where we included the physiological information, and we predicted that with the decreasing ice cover and increasing temperatures, the blooms of this diatom, this endemic diatom, will decrease, and they are already decreasing. So another interesting question is whether we can use these trait-based approaches to understand and predict harmful algal blooms. And this is something that I'm very interested and have been interested for a while. And so when we analyze different traits, we can isolate the traits of different HAB taxa and compare them to the non-HAB taxa. And so why do we need these trait-based approaches? So there are several questions that are still unanswered in HAB research. So for example, we know that HABs may be becoming more frequent in the future, at least in some parts of the globe. But still, it is hard to predict the magnitude and the timing of those blooms. So for example, Lake Erie, that has blooms of microcystis almost every year, those blooms vary in severity. And we still don't know why. Of course, it has something to do with nutrients, temperature, but we don't have the exact answers yet. Moreover, when we think about harmful algal blooms, we shouldn't just focus on microcystis because there are other HAB taxa that are important. For example, recently there are blooms described in Lake Superior, and those blooms consist primarily of Delicospermum, formerly known as Anabana. And in Lake Baikal, despite it being oligotrophic, we see blooms of, well, Spirogyra. It's not a toxic bloom, but it's a nuisance bloom. And we also see blooms of Delicospermum, which can be toxic. So when and where different HAB taxa would dominate. So looking at their traits may give us answers. So we can conduct comparisons of traits at different levels. So here we compared different groups. We compared cyanobacteria with other groups of algae. And so we found that growth rates of cyanobacteria are intermediate compared to other groups of phytoplankton. And we know that high growth rates are usually selected for under fluctuating or high nutrient conditions. And so cyanobacteria seem to be in the middle, although they thrive under high nutrients. So we also looked at the competitive abilities or affinities for phosphorus and nitrogen. And so cyanobacteria seem to have high affinity for phosphorus, but low affinity for nitrogen. But I have to say that this is maybe an artifact, because if you include nitrogen fixers, that basically throws off all the analyses, because nitrogen fixers do not need to have high affinity for nitrogen, as they can fix atmospheric nitrogen. So distinguishing between nitrogen fixers and non-fixers is really crucial when we analyze traits of harmful cyanobacteria. So then we looked at light traits, and we found that cyanobacteria are really good light competitors. So they have these high initial slopes of growth irradiance curves compared to other groups. And at the same time, they seem to saturate or get inhibited by lower light levels. So there is a trade-off. They're adapted to low light, but they're sensitive to higher irradiances compared to other species, other groups of phytoplankton. So when we looked at temperature, the situation was interesting, because when we look at temperate latitudes, the optimum temperatures and the maximum temperatures of cyanobacteria are higher than for other major groups of phytoplankton, such as green algae and especially diatoms. So, however, it is important to think about the latitudinal distribution of these traits. And so it turns out that in tropical systems or subtropical systems, the temperature traits among these three major groups are more similar. So that means that with warming, the stimulation of cyanobacteria, the preferential stimulation of cyanobacteria may be less likely than in temperate systems. So therefore, it's really important to get the trait information, not only for temperate lakes, but for lakes along the latitudinal gradient, and measure traits of different species and different isolates. So when we put these trait ranges together in a three-dimensional plane, for example, we find that these different groups of algae, so diatoms, cyanobacteria, and green algae, they do differ. So they are not on top of each other in terms of their trait values, but they're separated in this trait space, which is really interesting. And it means that some conditions, for example, that let's say early in the season, the conditions would be selecting for species with such a combination of traits, and that would primarily be diatoms. And later in the season, so the optimum trait combination would have a certain trajectory. And for example, later in the season, we may have the selection for a different combination of traits. And that would happen to be the trait combinations that are characteristic of cyanobacteria. So basically, this kind of a simple illustration how when you focus on traits and trait combinations, you can think what potential winners or losers would be under different environmental conditions, be it across lakes or in the same lake during different seasons. And so you can explain then, for example, seasonal succession in a lake, looking at traits. And so here is an example of different trait combinations. And you can see, so depending on what traits you're considering, you may have more differences or smaller differences across taxonomic groups. So when we compare traits, we can compare them across taxonomic groups, or we can compare them within taxonomic groups. So for example, within cyanobacteria, we can look at traits across different genera, or even within each genus, and we can have isolates and measure their traits. And what we've been finding, and other people have been finding it, is that there is a lot of variation, even within a given genus or a given species. And so, for example, this invasive toxic cyanobacterium cylindrospermopsis tends to have higher temperature optima than Microcystis. But there are some strains of Microcystis that showed really high temperature optima. So it may be that in the future, those strains would be selected for. So we really need to have this information where we compare traits not only across taxonomic groups, but we compare traits across species within one taxonomic group. And we can go further and compare traits within a species. And when we make these comparisons, we can ask different questions. For example, when we compare traits across different functional groups, we can ask the following questions. Is the cyanobacterial dominance likely? Whether the conditions that we see would be selecting for that combination of traits that is characteristic of cyanobacteria. And we can also ask, will there be a bloom of cyanobacteria? Then when we look at the differences across species within cyanobacteria, we may ask different questions. We may want to know what cyanobacterium would dominate. And in different lakes and different seasons, different hab taxa may dominate, and that is determined by their traits. And we can ask whether the hab taxon be a nitrogen fixer or not, and whether the bloom would be toxic or not, because within cyanobacteria, we have both toxic and non-toxic species. And we can ask whether there will be a succession within a bloom. So at that intraspecific level, we can ask questions whether there will be a change in toxicity within a species, single species bloom, because it has been shown that different strains, even within a single species, may differ in their toxicity. And then when we assess this intraspecific trait diversity, we may ask a question whether there would be a potential for trait evolution or evolutionary adaptation of a given species to changing environmental conditions. So I already mentioned that when we think about cyanobacterial traits, we should really be careful whether we look at nitrogen fixers versus non-nitrogen fixers, because there are many differences. And of course, the main difference is how these non-fixers and nitrogen fixers respond to nitrogen. And we found when we analyzed a large data set of phytoplankton dominance and cyanobacterial dominance in different lakes in the US, we found that there are different responses to nutrients such as nitrogen, for example, and of course it makes sense. So we found that only non-fixers were stimulated by inorganic nitrogen, and nitrogen fixers were not, which makes sense. So trait-based approaches can be used for a lot of different things. And for example, one thing that is being applied a lot to terrestrial systems and is starting to be applied to aquatic systems is to characterize the state of an ecosystem, whether it's a a pristine state or a disturbed state. So in how much of a stress a system is found. And so we can look at the community weighted trait means, and we can focus on different traits depending on our question. We can focus on temperature traits, we can focus on nutrient traits, we can focus on growth rates or sizes and so forth. And so, for example, one can envision such a scenario when we look at the distribution of a given trait of interest. We can find that in a pristine ecosystem, we have one trait distribution with a certain trait mean and a certain trait variance. And when we look at a disturbed ecosystem, we may find that the trait distribution shifted. So for example, we may find that the community has a different mean of a trait, or it has a smaller variance in that trait, which usually can be a proxy for functional diversity or functional health of an ecosystem, so the trait variance. So, and then we can sort of characterize an ecosystem based on the community trait distributions. And so then if we measure traits in an ecosystem, we can then determine how impacted or how stressed a given ecosystem is. So to summarize, there are some take-home points, and there are, of course, many more, and I have not talked about a lot of different aspects. But so we found that major phytoplankton groups tend to differ in their traits. And there is also a considerable intra and interspecific variation in traits. And this is really interesting, and it gives us information to determine what species may dominate and whether there would be an evolutionary adaptation. And so we found that traits can explain species dynamics and distributions, and we had a lot of papers on that, and other people are publishing on that. And so I say that the knowledge of traits is really important if we are to predict what groups of or what species will be dominant in the future under different conditions. And so we can focus on different aspects of this trait-based framework and characterizing, for example, better this intra and interspecific variation in key traits is something that needs to be done. And we can measure traits either in the lab, in the field, or we can derive traits using various techniques from the environmental distributions of different species. And then looking at stressor interactions and how traits may change depending on those interactions is another frontier. Then thinking how traits can evolve, because of course they are not fixed in space and time. And the potential for trait evolution is really important for our predictive capabilities. And then another interesting frontier is to look at the underlying genomic characteristics of phenotypic traits. And that could allow us maybe not to measure phenotypic traits, but just to look at genomes of organisms and to be able to predict what physiological or ecological traits that they would have. And another frontier is that we still don't know what traits are important in determining intertrophic interactions. For example, predator-prey, well, we know more about that, but parasite and host interactions, we still don't know many of the traits that may be important. So this is another fascinating frontier. So I will stop here, and I also want to mention that I'm looking for a postdoc with strong quantitative skills. If you're interested in in these approaches, trade-based approaches to aquatic ecosystems, please do get in touch. And thank you very much for listening.
Thank you so much, Elena, for that presentation. I think it was a very good introduction and overview about all the possibilities that trade-based aquatic ecology is capable of. So thank you for summarizing all of that. Let's see if we have any questions and open it up to the participants over here. So if you have any questions to Dr. Litchman, please go ahead and either write it in the chat or just ask directly. So I will wait a couple minutes here. Okay, well, no one's jumping right now, so I actually have a question for you. So I was interested, you had that slide where you compared, there was a difference correlation between the marine and the freshwater environment. And I was wondering if using trait-based ecology, you can maybe tell us other examples where there were either contrasting, results regarding to these two major aquatic realms or comparisons, so where we're the same.
Yeah, it's a great question. I've been fascinated by comparing the two environments, and I've worked in both, and it's actually informative. And I would say, so some of other examples is that diatoms seem to behave differently in marine and freshwater environments. So in freshwater environments, many diatoms, and of course I have to stress the fact that diatoms have a wide interspecific differences, like range of traits. So, but in general, it seems that in freshwater environments, diatoms are maybe adapted to sort of lower nutrient conditions compared to, let's say, green algae or cyanobacteria. And they don't have necessarily the highest growth rates. And in marine environments, diatoms seem to be occupying a somewhat different ecological niche compared to different groups of phytoplankton. Marine diatoms are known to thrive in really high and fluctuating nutrient conditions. And so they have extremely high growth rates compared to other marine groups. So there are these differences. And also I've been fascinated comparing the strategies of harmful algae in marine and freshwater environments. And again, it seems that there are differences. So for example, if we focus on dinoflagellates that are very important toxic algae in the marine environments, and freshwater cyanobacteria that are like key players in among the HAB taxa in freshwaters. Again, there are different strategies there. So it's like, why do we have these differences? So like kind of naively, one could have thought that all harmful algae are kind of similar in both marine and freshwater environments, and they occupy similar niches, but it's not the case. So it's like, it's really fascinating. Yeah.
Yes, thank you so much. Those are pretty interesting comparisons. So I think we have two questions here, maybe related to this question. Let's see. The first one hand was from Tom Shapwell. Would you like to ask your question?
Hey, Tom, great to see you.
Yeah, you too, Elena. Thanks very much. I really enjoyed the talk.
Thank you.
Yeah, so my question is basically, what role do you think organisms have that have like just an intermediate trait value on the gradient? They're always less competitive than some, but more competitive than others. And if I would try to model this, they would usually become extinct in the model sooner or later, which could be just because the model is too simple. So I was wondering if you have any insights on this question.
Wow, that's a great question. And I I do not have any earth-shattering insights, but I think maybe so that those species are intermediate. It either can be that maybe in some traits they are not intermediate and we're just not including them, or it could be sort of like these are like sort of transition species, you know, they're selected for maybe during the times of transitions. And one question that I'm really interested in is that when we look at responses of communities or ecosystems to extreme events, whether we expect to see species with extreme traits, right? So thriving. Let's say if we have a heat wave, would we expect to see probably species that are really warm adapted to thrive? And those intermediate species maybe are sort of out of luck. Who knows? Yeah, but this is really interesting. It's sort of like this these different ecological strategies. Maybe it's the sort of being in the middle is not that bad. And yeah, I'm curious whether models could help, like could sort of be made that those species persist. And one way would be like adding some immigration, maybe, you know, like to those species. But yeah, this is fascinating to think about different ecological strategies with respect to trait values. Yeah.
Yeah, thank you. It's a little bit my feeling as well that our models just don't have enough dimensions to come anywhere near the complexity and can represent that many traits or trade-offs. Yeah, thanks a lot. That was really interesting.
Thank you.
Okay, so we have the next question from Natalia Vargas.
Hello all, and nice to meet you. Thanks for the conference. It was very interesting.
Thank you.
I am pretty new in this topic, so I have a question that is in my mind all the time. With this ecological trait approach, and also like with the meta barcoding things that are kind of new in some ecosystems, but I am thinking like, what is the role of the taxonomic identification and how this can like couple with these new approaches? That is like one first question, and then I have another one.
All right, so yeah, I'll answer this question. So it's a great question because, you know, some people say, well, focusing on traits is good and fun and it gives you a mechanistic understanding of the processes, but still I'm interested in a certain species, right? And I think it's kind of, I'm a phytoplankton fan, you know, and I care about individual species, not only traits. But I think we should really retain both the trait information and the species information, right? Because like if we predict, okay, just looking at traits, a small round species with good nutrient competitive abilities will dominate. This is not the most satisfying answer, right? So we gotta go to like species, like we have to have some taxonomic resolution there. And I think like sort of both looking at traits and taxonomic identification is interesting. So.
Thank you. So it's like the ecological traits approach doesn't like take away the taxonomic identification, right?
Yeah, but so like, for example, if we want to predict what species would be dominant under what conditions, we have to look at its traits, right? Because like we just can't like say, okay, let's say Senadespas will be dominant, but why? We got to know its traits. Yeah.
Thank you. And I also have another question about if you know if there are works of trait ecology on periphyton.
Yeah, great question. So like in the beginning, I gave like sort of like the example of the ISI search, you know, of the papers. And I I tried looking at periphyton, and there are many fewer works, and we know very little about periphyton traits. And this is something that I think researchers should focus on as well, because, for example, you can have maybe very similar, let's say cyanobacteria, but they may have different, very different traits when like it's a planktonic versus periphytic cyanobacterium. Yeah, we know very little, I think.
Okay, thank you very much.
Mhm.
All right, great questions here. Next one, Thomas Shannon, please.
Hello. Thank you so much for your talk. I found it really insightful.
Thank you.
So I was thinking about the fact that most of the traits that you're describing are traits that are reactive to the environment, either the nutrients or the biotic conditions like grazing. But that some of these traits like toxin production are things that are being contributed to the environment. And so I was curious if you've thought much about the ways in which you would be able to expand a trait-based approach to see how the community is influencing the whole ecosystem as an ecosystem function rather than as a response to it.
Yeah, it's a great question. And terrestrial plant ecologists especially, they seem to sort of divide traits into response traits. That's what you said, and that's what I'm mostly focusing on, and effect traits. So basically how different traits mediate the effects of organisms on the environment. And I agree that sometimes this separation is useful, as in your example, like toxins, for example, but sometimes they sort of they blend together. For example, nutrient utilization traits are both the response traits, so they determine how a certain organism grows under different nutrient conditions, but at the same time, it would be affecting nutrient concentrations in the environment. So it's kind of an effect trait as well. And the same with nitrogen fixation as well. And I agree that we should sort of not only focus on how different organisms respond to the environment, but how they affect the environment. And then maybe tying the concepts of, I don't know, keystone species or whatnot, and how different traits affect ecosystem functioning. And this is actually something of interest to me, and I'm sure many other people, how to incorporate traits into sort of global ecosystem models. And I think this is like another frontier that I did not list, but it's definitely a frontier. Yeah. Thank you.
Thank you.
Okay, we have another question from Mark Gessner.
Hey, Mark.
Hello, Elena. Good to see you. If we have time, I'd be happy to ask a question relating, very general, relating to your statement that you want to reduce complexity by working on traits rather than taxa. And I was wondering whether you could expand a bit more on this, because I can also lump taxa together. You made the point that there are different diatoms that have different functionalities. But isn't it ultimately a matter on how finely I separate either my traits or my taxa, how much complexity I have? And the difficulty is, of course, to find the right level to answer your question. But I was wondering whether you think it's a really fundamental difference or just another way of approaching things.
I think, so one example I can give you is that, for example, if you think about how different traits are related to each other, we can, let's say, have functions that describe how size affects nutrient uptake, for example. And then we can really reduce this trait dimensionality even, we can have a relationship where we plug in size, and then we don't need to measure the nutrient uptake rates, for example. So we already have this relationship. So it's kind of like reduces this complexity of like how many traits we have to include explicitly by sort of measuring the relationships among traits, for example. So
Sure, but you could do that by saying green algae, you know, tend to have high nutrient requirements or have higher growth rates or things like this.
yeah, but you still need to sort of like, and then we want to know how they take up nutrients. And then if we put in size of them, you know, we can have this relationship. If we just say green alga, like, so how does, what nutrient uptake values does it have?
Mhm.
So, but if we put in this trait size, then we can really immediately derive nutrient uptake capabilities. So,
Would you want to combine that with the taxonomic information?
yes, I think it's important because we found that for some trait relationships, it really matters what taxonomic group you're considering. For example, we measured grazer resistance as a function of cell size, and it turns out that it is different for cyanobacteria versus green algae. So, and I don't know for like what the mechanisms are, maybe toxicity, maybe some mucilage, maybe something else. But yeah, so I'm not saying that we should remove the taxonomic resolution there. I think combining the two would be the best.
Yeah, sure, thanks. Yeah, in relation to Natasha, was that her name? Question, if we identify the species by molecular tools, then we could also, and there are relationships between the genes and the traits, then we can actually get around the taxonomic information.
Exactly, yeah. So for example, how many nitrogen and phosphorus transporters this genome has and how well it could be related to nitrogen uptake or like nutrient uptake capabilities. Yeah, and there are such studies and sometimes it works, and especially for bacteria, for example. Yeah.
Thanks very much.
Thanks, Mark.
Okay, I do not see more questions. Anyone? Last call for questions here?
And of course, feel free to get in touch with me, and I'm happy to answer your questions or doubts about trait-based approaches.
All right, well, I think we do not have any questions right now, but I'm sure they will get in touch with you via email or visiting your website, basically. So thank you so much again for the presentation. We very much enjoyed your talk, and the recording will be shared afterwards with the participants who have registered. So thank you so much for your time, and thank you for being here, everyone.
Thanks, everyone, and thank you for inviting me and organizing this.